So now it's we have told you a number of algorithms or at least a way to think about these problems. We want to show you some scenarios if you have a collaborator or if you're dealing with a project, how you think about it, okay? So let's think about this first scenario. So we have a transcription factor X, which is very important in cancer. In this one cancer type. And in this cancer, people have done a lot of profiling of this cancer type, say lung cancer or some, non-small cell lung cancer. Let's say you have expression data, but let's just say it's very difficult to do ChIP-seq for this transcription factor directly in the tumors. Therefore, your collaborator started by doing a ChIP-seq of this factor in a cancer cell line Y. Okay, so very often you have a cancer cell line. And they also have this RNA-seq for the same, in the same cell line in two conditions. One, when this TF is active, and in another condition when you knock out the factor, when the factor is inactive. And so you have two expression data in the cell line, one ChIP-seq data in the cell line, and a big tumor cohort. And they come to you, say, oh, well, you took this STAT115, 215 course now. What would you do with this data? Can you tell me? How do you start with? Yeah, you can just say it out. Yes, very good. You QC your ChIP-seq, make sure that they did a good experiment, right? You can check how many peaks they're getting. Are they in the DNA's hypersensitivity regions? Are they conserved? Do you see some motifs, right? That's important. Then what? Sure. That's in the first module, right? You make sure, yeah, usually the RNA expression is okay. Yeah, so one thing you might want to look at is you look at the motif of the transcription factor ChIP-seq. You ask, is it collaborating with some other factors that might be important? Or you can just do this ChIP-seq peak in Cystrome to see whether other factors have significant overlapping binding sites, which also tell you they might be interacting. If you are able to identify a collaborating factor, is there some, so actually at this time, you can talk to your collaborator and say, another factor is very important too. You didn't know, maybe, but that's also important. And so they might want to do some experiment on that. In fact, this is what we did in the first transcription factor ChIP in human. It was actually our study where it's the exact same situation. We knew estrogen receptor was important in breast cancer, but it was impossible to do breast cancer ChIP like 15 years ago. And so we did the ChIP in a cell line. And once we identify motif, we say, oh, turns out that there's another transcription factor that's important. And our collaborator actually knock out that transcription factor and found, oh, it is indeed important. When you knock out the transcription factor, estrogen receptor cannot bind to those sites anymore, right? So you can use it to identify its important friends or collaborators. What's next? Yeah, you can do differential expression on RNA-seq. And then we want to probably identify its target, right? So there you use all the binding sites. You have like this regulator potential to help identify which are its putative target. But also you look at those genes that are also differentially expressed, then they will give you the target list. So remember here, each transcription factor, in terms of its target, it's not a yes-no binary decision. It's like a very strong target versus those weaker target. It's like a gradual, right? But you can get, say, the strong ones, get a few hundred of those targets, and then you can run gene ontology, David or gene set enrichment, see whether they are related to the disease. What other things can you do? Yeah, so sometimes you can also look at, maybe when TF is interacting with, so your TF X is interacting with transcription factor A it regulates some target. But when it's interacting with a factor B, it might have a slightly different function. And from this, yeah, you might want to use, like, so there, I don't think there are like very standard tools for doing this, but you can imagine for TFX, you can get, the TFX with also motif A, with motif B and motif C, and you can see maybe they have different target genes as well. But remember this study we did in cell lines, right? We want to somehow bring it back to the tumor as well. Is there something you could do to go back to your tumor expression data to ask, is my cell line giving me useful information? Can you do something there? So first thing you probably want to check is, in this tumor data, you might have a cancer and adjacent to normal. And you can ask, among these targeted genes that we see in the cell lines, are those genes also differentially expressed in the tumors? Right, and then you see whether it's relevant. But yeah, also, as the student mentioned, for those tumors that have either high expression of the TF or the high expression of its target genes, do you see the patient have different survival, right? Is this factor really important for that disease? Whether the target genes tell you that this factor is indeed very important, and what's the pathway, what's the gene that really caused those differences? Okay, so that, by this time, you can draw the figures and start writing the paper, right? That's the idea. Okay, so second scenario, there is a developmental process, and we don't know what factors are involved in this regulation. Let's start from very simple. So you can see actually, when you have to deal with gene regulation, very often we work very closely with the experimental biologists. Sometimes we can help them design an experiment. They actually look at the result, or we help them analyze, we generate new hypotheses. They will go to do more experiments based on what we see. Can you think about, so your collaborators, I'm interested in some like stem cell differentiation, and they might have very little cells available, and they don't know what factor is important. What do you do? If you have cell lines, that would be great, but maybe you don't. Let's just say you have an embryo, little few cells. Maybe you get 1,000 or 10,000 cells, which is usually not enough to do a transcription factor ChIP-seq. But you can do RNA-seq on those 100 cells, like 10,000 cells you can do, right? You can also do ATAC-seq now, or you might be able to do some histomark ChIP-seq. In terms of understanding what transcription factor might be important, I would say ATAC-seq and K27 or acetylation ChIP-seq are the most informative ones. DNase is also extremely informative, but you can't do DNase in like 1,000 cells. It's just very hard, okay? So ATAC-seq is easy, RNA-seq is easy. You can do those in, maybe 1,000 or 10,000 cells. You get the expression, you get the collection of all the binding sites. What do you do next? So you make a recommendation, your collaborator did this assays. Now you have a bunch of FASTQ. What do you do? Yeah, you QC the raw data, right? You do your read mapping. For RNA, you can do the RNA-seq. For the ATAC-seq, it's like you can use the similar ChIP-seq type of QC to QC your ATAC-seq. Then what?
Do you have this different time points? Or just one time point?
2 times, X and Y, 2 time points, right? So it's like before differentiation and after differentiation. You have 2 states.
You start calling that.
Exactly. Excellent. You look at the, so you do differentially expressed genes. You can also look at differential peaks as well, right? The ATAC-seq peaks. And based on based on the differential peaks, then you can look at motifs. Or you can use those differential peaks to do overlap with public ChIP-seq data to ask what other factors bind to those differential peaks. Right? That will tell you, oh, some factors might be important. Then you look at those differentially expressed genes, or at least this gene needs to be highly, or this transcription factor needs to be highly expressed in the cell you're interested in. And that factor might be important in this differentiation process or the developmental process, right? And then based on the, so there that actually depends, you might need to look for some tools. They may or may not be completely available. You might have to write some code. Based on those differential peaks and also the differentially expressed genes, you might be able to get the correct TF and also those target genes for those TFs. Then, you might want to do some experimental validation. Say, if I knock out this factor, does the cell still differentiate? Right? You can see. Or if you knock out one cell, it automatically differentiates, or you overexpress some transcription factor, it automatically differentiates. By the way, once you have the target genes, you can again run gene ontology, different like a gene site enrichment, right? That's another scenario. A third scenario is from GWAS study, you spend a lot of money, you got a single GWAS SNP. What do you do? This SNP X in disease Y. Using SNP arrays, what do you do? Okay, you can try that. What if it doesn't have any peaks overlapping? Exactly. Yeah, that's the idea. You find the SNPs in LD with this X. Then you ask whether there are any public, ChIP-seq or ATAC-seq or DNase-seq that overlap with those SNPs that in LD. I think, if you're lucky, the SNP lands right in the peak. Otherwise, it could be in LD. Then what do you do? Sam? You may or may not have the information. So supposedly you have, you can look at the EQ, like the GTX data, which has, all the different tissues, or the HAP map data. You could try, like some EQTL analysis, right? You ask, or you can look at the ChIP-seq raw data to ask, if this region has the SNP, does that change the peak signal in that location, in that peak, right? When you have the AA allele or AG allele or the GG allele, does that change the peak signal in that location? And then you try to see, when you have a difference in the peak signal, is that correlated with the gene expression, using either EQTL, or you can ask, does existing, high C data or some, high ChIP or whatever available chromatin interaction data, where does this SNP link to in terms of potential target gene? You could, if you know what transcription factor, you can look at motifs. You can use, I guess you can look at the motifs to see what motif binds to this region, whether the SNP will perturb the transcription factor binding. And if you know the transcription factor that binds there, that can also help you identify the downstream target, because you can use not just that SNP, you can look at the ChIP-seq data for that transcription factor to get its putative target genes, right? So the target gene can come from EQTL, can come from chromatin interaction, can also come from ChIP-seq for the transcription factor. And then depending on the target gene, maybe you want to see whether the target gene is related to the disease, some known disease association, or some cancer, survival differences. Okay. And then maybe they will do some experimental validation. So for example, if they delete this region, or they change the nucleotide in that base, does that change the binding? Does that change the nearby gene that you predicted? Right. So then they have a paper. So next scenario is supposedly we did a study of COPD or some whatever disease. You sequenced whole genome sequence of 100,000 individuals, and they did not see, I mean, for these, probably it's not a very strong p-value, but they just saw that there are like 1,000 rare variants in the disease case, which they didn't see in the normal. And they don't know which of these rare variants are real, but just they have a whole bunch that they see in a disease case. I'm like, what happens, right? So nowadays, I think in the precision medicine field, when they do whole genome sequencing, yeah, they just have a lot of differences so like that are observed in the case, but not in the control. And they might appear in 1 patient, 2 patients, or 3. And what do you do there? You have a lot. What do you do? Probably none of them very significant. They want to ask which ones are significant, which ones are potentially causal, right? They don't know. They just saw, okay, it appeared 3 times in the patient, but 0 times in the control. Is this significant? I don't know. Yeah, they definitely have this issue. So that's why all of these current precision medicine cases, people are starting to look at epigenetics to answer these questions. But if you were that collaborator of the precision medicine study, how would you go about this? Yeah, so you could, yeah, so if it's a case, yeah, so case control, yeah, of course you want to get the most significant ones, right, the more power one. The rare ones may or may not be important, we will see. But how do you give some hint which of those SNPs might be important? Power is, yeah, your p-value, power analysis is one. What else? Yeah, the location, but how? Yeah, the first is you filter. If it's, so here you don't need to look at LD anymore because if it's rare, it's because you did whole genome. The reason we have to look at LD before is because we were starting with a SNP array. The causal SNP may not be on the array at all, but when you do the rare variant, when you sequence everything, that SNP or that rare variant it needs to be on the DNA's peak. Of at least in hundreds of DNA's data, at least it was in a peak sometimes. If it's never ever in a peak, you probably can eliminate those. They're pretty hopeless, right? And then, yeah, so you ask, okay, out of all those SNPs or the rare variant that overlap with DNAs, which is the cell type that has the highest overlap? So supposedly you start with 1,000 rare variant. When you use filter by DNAs, you have only 100 left. 900 are gone, totally hopeless, right? The 100, you ask out of the different DNA's data, which one has the best overlap with those 100? That can probably tell you what cell type it is. So interestingly, when people are looking at the disease related to neuronal, to some cancer, to other things, actually the neuron disease may not really be a neuron DNase. It could be related to the immune system. Actually, a lot of disease now people find, oh, it's related to the immune system. And so you want to find out what cell is really relevant. So maybe out of those 100 SNPs that are still in DNAs, you ask, which DNA's data give me the best overlap with all those 100? Okay, those are the cell lines which probably are relevant to this disease setting. So help you identify the cell lineage. And once you have that DNA's data, you roughly also know what genes are probably active in that cell, because that gene promoter will be active. Right? You know, oh, okay, this gene at least is promising. And then you look at motifs. In those regions, you ask what ChIP-seq might bind there. Would that SNP or the variant perturb the transcription factor motif or the transcription factor binding? If you have ChIP-seq data, you might also see, oh, if I have a C versus T or whatever, A or G, does that change the signal? right? You can see whether there's STAT-specific or allele-specific peaks signals. And then you might want to find the nearby putative target gene. If you know what transcription factor binds there, you might be able to find its target gene based on the ChIP-seq data. You might also look at the chromatin interactions, right? And also, you want to make sure that gene is at least active in the cell context you're looking at, because the DNA's data tell you the lineage. You will know, okay, that's the target, not the nearby, the closest gene, which is not even active in that cell type. Right? So that's some of the ways you potentially can look into this. And of course, these rare variants are kind of all over the place. At the end, you ask, okay, out of those putative target genes, are they known to be linked to some disease? So actually, I just searched this. There's a database for this. It's called Disease Genes Net. Yeah, so supposedly the 100 SNPs that land in DNAs is mapped to 50 genes. And turns out those 50 genes might be related to a disease. Okay, then you found something, right? So that's a potential approach. So anyway, actually, our lab is working on a lot of these related things. Because I think the simple stuff, QC, ChIP-seq, target, those are a lot of those are done. So we are working on a lot of these potential things to hopefully provide computational solutions to this. That's why it's important area. So for all of this, you can see getting the motif is important. Knowing which transcription factor binding there is important. Knowing all the binding sites for those TF in different cells is important. Finding its target gene is important. Right? So all of this takes additional research questions. Okay, but that gives you some ideas.